Picture this. It's the year 1683.
Okay, I'm picturing it.
So a Dutch scientist, Antony van Leeuwenhoek, is peering through this really rudimentary handcrafted microscope.
Right, the early days of microbiology.
Exactly. And he's looking at a sample of his own dental plaque.
Nice.
Yeah, a little gross. But right there in the fluid, he sees them, these tiny moving organisms, and he calls them animalcules.
Animalcules. I love that word. It's so archaic. But, it represents this massive turning point, the exact moment we actually laid eyes on the microscopic world.
It really was. And fast forward to today, we know those animalcules aren't just, hanging out in our mouths.
Oh, definitely not.
Our entire gastrointestinal tract houses just trillions of these microbes. It's this bustling ecosystem, the gut microbiota.
And we are really just now realizing that it basically holds the keys to our systemic health.
Which brings us to the mission for today's deep dive, because we are unpacking a massive paradigm shift in modern oncology.
A really massive one.
Yeah. We are looking at exactly how the gut microbiota acts as the master controller of this revolutionary cancer treatment called immune checkpoint blockade, or ICB.
Right. And to give you a quick baseline, usually we think of cancer treatment as a two-player game. Right? You have the pharmaceutical drug and you have the patient's immune system.
It's the drug in the body.
Exactly. But ICB therapies, which are drugs like anti-PD-1 or anti-CTLA-4, they work by cutting the brakes on your immune system.
Because normally those checkpoints stop the immune system from attacking healthy tissue, right?
Yes, but cancer hijacks those brakes to hide. So the drug cuts the brakes, which unleashes your cytotoxic CD8 T cells to go destroy the tumor.
But the wild thing we're learning from the source material is that there's a third player. If ICB takes the foot off the brake, the gut microbiome is basically providing the steering wheel and the gas pedal.
That's a perfect analogy. Because if that microbial communication loop is broken, the therapy just doesn't work.
And these bugs can be our best friends or our absolute worst enemies. Let's start with the bad actors. I read that 15% of global cancers are linked to chronic infections.
Yeah, it's a huge number. And the research points heavily to specific strains of E. coli.
Which we usually just associate with, terrible food poisoning.
Right, but in oncology, certain E. coli strains produce this potent genotoxin called colibactin.
Colibactin, and what exactly does that do?
So colibactin doesn't just irritate the tissue, it actively alkylates our DNA.
Wait, it alkylates it, it chemically alters it?
Exactly. It attaches these chemical groups right to the DNA structure. That causes profound genetic instability, which actively promotes tumor growth.
That is terrifying.
It is. And other microbes take a different route. They trigger chronic inflammation by prompting the gut to release inflammatory cytokines, like interleukin-23.
Okay, IL-23.
Yep, which then cascades into interleukin-17, and that just creates this highly inflamed environment that inadvertently feeds the tumor.
But then, on the flip side, you have microbes running, like counter-ops. The sources highlight Lactobacillus reuteri.
Oh yeah, the good guys.
Right. And this one produces a compound called reuteri, which essentially weaponizes chemical stress against the cancer.
It's fascinating. Reuteri increases reactive oxygen species, or ROS.
And that hurts the tumor.
Yeah, it actively restricts colorectal cancer by shutting down the tumor's ability to synthesize proteins. It's literally a microscopic turf war down there.
And understanding that turf war leads us to this huge clinical paradox the researchers found. So they looked at patients who took antibiotics right before or during their ICB cancer treatment.
And the outcomes were just staggering.
They really were. Those patients had significantly shorter progression-free survival and overall survival compared to the ones who didn't take antibiotics.
Which, if you think about it, makes total sense. Antibiotics are like clear-cutting an old-growth forest.
You're not just killing the weeds, you're destroying the whole root system.
Exactly.
Yeah.
But here's the real curveball. The data showed that late-stage melanoma patients who took over-the-counter probiotics also had reduced therapeutic responses to ICB.
Okay, wait. I have to pause here. Because this completely flips conventional wellness wisdom on its head. Probiotics are supposed to be, universally good for gut health. Why are they ruining advanced cancer therapies?
I know, it sounds crazy.
Yeah.
But it comes down to ecological diversity. OTC probiotics usually just deliver a massive concentrated dose of one or two generic strains, like a common bifidobacterium.
Right, you see those on the label all the time.
Exactly. But when you flood a complex ecosystem with millions of identical microbes, they can actually aggressively outcompete your diverse natural community.
So it becomes a monoculture.
Yes. And the data proves you need a sprawling, diverse metabolic network for the drug to work, not just a generic pill monoculture.
Okay, so let me just summarize this first segment for you real quick so we keep it all straight. 1, ICB therapies release the brakes on your immune system to fight cancer. 2, your gut microbes can either directly damage your DNA with stuff like colibactin, or they can kill tumors with Ruterin.
Spot on.
And 3, wiping out your gut diversity with antibiotics or flooding it with generic probiotics actively sabotages your cancer immunotherapy.
That's the perfect recap.
So if a generic monoculture ruins the therapy, what does the ideal gut look like? Who are the MVP microbes actually doing the heavy lifting?
The clinical data zeroes in on a few VIPs. The heavy hitters correlated with ICB success are Akkermansia mucinifera, Enterococcus hyrae, and specific diverse bifidobacterium species.
And we know exactly how they operate, right? They aren't just sitting there. They actively talk to our immune scout cells.
Right, specifically conventional dendritic cells, or CDCs.
Okay, so how does that conversation work?
Well, the CDCs are constantly sampling the gut environment. When they detect bugs like Akkermansia, it triggers them to pump out interleukin-12.
IL-12, and that acts like an alarm.
A massive systemic alarm. It dramatically increases the recruitment of those cytotoxic CD8 T cells, pulling them out of circulation and driving them directly into the tumor.
And it arms them too, right? Because the microbes boost the T cells' production of interferon gamma.
Exactly. IFN gamma is the primary weapon the T cells use to shred the tumor.
That is amazing. And the biological proof for this is arguably one of the most wild experiments I've ever read about. The fecal microbiota transplantation, or FMT studies.
Oh, this was a landmark moment in oncology.
Walk us through it.
So researchers took stool samples from human melanoma patients, half from patients who responded beautifully to ICB, and half from patients who were totally resistant to the drug.
Okay.
Then they transplanted those human samples into genetically identical mice that were raised in completely germ-free environments.
Wait, totally germ-free, like zero endogenous gut bacteria.
Blank slates, zero bugs. So they give these mice tumors, treat them with ICB, and the response perfectly mirrored the human donor.
That is insane.
I know. The mice with the responder stool fought off the tumors. The non-responder mice failed. The therapeutic response was literally inherited via the microbiome.
That fundamentally changes what a therapy even is. We're transplanting an ecological state. And they're doing this in humans now, right?
They are. Clinical trials have successfully used FMT from ICB responding humans to cure resistance in non-responders with melanoma, renal cell carcinoma, and non-small cell lung cancer.
Okay, mini recap number 2 for you. Specific microbes like Akkermansia are the true MVPs of cancer therapy.
Yes, they are.
They work by boosting IL-12 and IFN gamma, which supercharges your T cells. And most importantly, this ICB responsiveness is completely transferable via fecal transplants.
Exactly right.
Okay, but this leaves us with a glaring geographic problem.
The location issue.
Yeah. How does a microbe sitting in your colon fight a melanoma on your shoulder or a tumor in your lung?
That is the million-dollar question. And researchers have organized the mechanisms into a great memory tool, the TAM framework. TAM.
Translocation, antigens, and metabolites. Let's hit translocation first, because normally the intestinal wall is a fortress. Bacteria stay in the gut.
Normally, yes. But a fascinating side effect of ICB therapy is that it makes that gut barrier slightly permeable.
It causes a mild leakiness.
Yeah, so specific microbes physically cross the barrier. They get picked up by those dendritic cells and carried into the mesenteric lymph node, or the MLN.
And lymph nodes are basically the immune system's military academies.
Right. So by showing up there, these microbes act as an endogenous adjuvant. They prime the naive immune cells systemically before they patrol the body.
That makes sense. Which brings us to the A in our framework. Antigens, or molecular mimicry. I love this concept.
It's basically a case of lethal mistaken identity.
Because an antigen is just a structural tag, right? A shape the immune system uses to recognize the threat. So what happens when a bug and a tumor have the exact same shape?
Well, researchers found a specific bacteriophage, which is a virus that infects the Enterococcus bacteria in your gut. And this phage has a molecular tag that is structurally identical to an antigen on certain tumor cells.
Okay, but wait. If they look identical, isn't there a risk of friendly fire? Like, does the immune system attack the gut instead of the tumor?
That is a huge friction point, and we'll talk about autoimmune risks in a bit. But for clearing the cancer, this mimicry is incredibly beneficial.
Because it primes the pump.
Exactly. The immune system mounts this massive response against the gut microbe. But as those prime T cells circulate, they bump into the tumor. They see that identical structural tag, lock on, and obliterate the cancer.
It's like using the gut bug as a skeleton key to unlock the tumor's defenses.
That's a great way to put it.
Okay, so that's T and A. Let's move to M, metabolites. This is where the chemistry really gets intense.
Because bacteria are just hyperactive metabolic factories. They digest our food and excrete chemical byproducts or metabolites that cross into our bloodstream and act like systemic drugs.
And the most famous ones are short-chain fatty acids, or SCFAs, like acetate, propionate, and pentanoate. We know these boost T-cell function. But the source material threw a massive curveball about one specific SCFA.
Butyrate.
Ah, yes, the butyrate paradox.
Because I've always heard butyrate is the ultimate gut health hero. Why is it suddenly a villain here?
It just shows how biology is rarely black and white. Systemic butyrate is great for general mucosal health.
Yeah.
But during anti-CTLA-4 immunotherapy, high butyrate is detrimental because it specifically upregulates regulatory T cells, or TREGs.
And TREGs are the peacekeepers. Their whole job is to stop immune attacks.
Right. So you're giving a drug to unleash the immune system, but you have a metabolite that is hyperactivating the peacekeepers.
You're slamming the brakes right back on.
Exactly. It creates therapeutic resistance.
That is wild. And there are other crazy metabolites too, like inosine.
Oh, inosine is incredible. It's produced by bifidobacterium. It circulates and binds to adenosine 2A receptors directly on the T cells.
And that gives them stamina, right? Yeah, it's like a sustained release energy reserve, so they can keep attacking a tough tumor.
Then there's indole-3 propionic acid, or IPA, made from dietary tryptophan. The epigenetic stuff here sounds like science fiction.
It really does. IPA travels to immune cells and alters an epigenetic marker called H3K27 cytylation.
Okay, break that down for me.
Basically, it physically unspools certain regions of the T cell's DNA. It unlocks genes that convert the cell into a more stem-like state.
Meaning they don't just fight and die, they can self-renew. Infinite ammo.
Exactly. And then you have TMAO, which induces pyroptosis in tumor cells.
Pyroptosis? That just sounds violent.
It means fiery cell death. It's highly inflammatory. The cancer cells rupture, spilling their hidden antigens everywhere, which just compounds the immune attack.
Wow. Okay, and the last one we have to cover because it blew my mind. Secondary bile acids.
Oh, the hormone blockers.
Yes. So the liver makes primary bile acids, the gut bugs eat them, and turn them into secondary bile acids, specifically one called 3-oxodelta-476-LCA.
Right, and this metabolite actively antagonizes the host's androgen receptors.
Which is huge because some tumors rely on androgen hormones to grow. So the bacteria are literally running a compounding pharmacy to starve the tumor of its growth hormone.
It's beyond elegant.
Okay, mini recap 3. TAM translocation primes the lymph nodes. Antigens create a lethal case of mistaken identity. And metabolites like SCFAs, inosine, and bile acids act as systemic drugs to alter immunity and starve tumors.
Perfect. But, if metabolites are the key, we have to talk about the supply chain. Where did the bacteria get the raw materials to make this stuff?
Diet. Diet is the absolute architect of the microbiome.
No.
And the data on dietary fiber is just unequivocal.
Oh, absolutely. A high-fiber diet feeds keystone bacteria like Faecalibacterium, which drives the mass production of those beneficial SCFAs.
But then you have the standard Western diet, low fiber, high fat.
Yeah, so you starve the gut of complex carbs, and the ecosystem completely shifts. The SCFA producers die off, and bugs that digest animal lipids take over, producing highly inflammatory metabolites instead.
Which brings us to the obesity paradox. This is a huge debate in oncology, because normally diet-induced obesity increases tumor risk and impairs T cells. But some clinical data shows obese patients actually have better outcomes with ICD. How is that possible?
It's heavily scrutinized, but the theory centers on mutational burden and baseline inflammation. Obesity creates a state of chronic hyperinflammation, and that metabolic stress can cause the tumor to mutate rapidly and erratically.
Making it sloppy and highly visible to the immune system.
Exactly. So when the ICB drug cuts the brakes, the immune system spots this massive number of mutations and mounts a hyper-aggressive attack fueled by all that baseline inflammation.
So it's not that obesity is strictly protective, but rather this highly mutated, inflamed environment just reacts explosively to the drug.
Right. It's incredibly nuanced and depends heavily on the type of dietary fat and the specific microbial shifts.
Okay, quick recap 4. Diet shapes your gut's metabolic arsenal. High fiber equals better ICB response. And obesity has this highly complex, sometimes paradoxical relationship with immunotherapy that we are still figuring out.
Exactly. But all this explosive immune response brings us to a massive double-edged sword.
Right, because if you take the brakes off the immune system, it doesn't just attack the tumor.
No, it frequently attacks healthy tissue. In clinical terms, we call these IREs, or immune-related adverse events. Autoimmune conditions like colitis, which is severe gut inflammation, or nephritis in the kidneys.
It sounds devastating for a cancer patient, but there's a clinical silver lining, right?
Yes. Developing mild IREs is actually a strong positive indicator. If you get a mild autoimmune rash, it proves the drug is working. The immune system is unleashed, which usually means it's destroying the tumor too.
But ideally, we want to kill the tumor without destroying the patient's gut. And the microbiome data suggests we might be able to decouple those two things, because different bugs cause the toxicity.
Precisely. Like a high abundance of Streptococcus bacteria is heavily correlated with dangerous IRE toxicity.
But researchers are going way beyond just looking at species now, right? They're using machine learning to look at the metabolic pathways.
Yeah, and they found a massive signal with vitamin K2.
Okay, tell me about that.
The machine learning models showed that the biosynthesis pathway for menaquinone, which is vitamin K2, is robustly linked to reduced autoimmune side effects.
How does a vitamin stop the toxicity?
Well, vitamin K2 appears to actively suppress a major inflammatory signaling pathway called NF-kappa B. So if your gut is rich in bacteria making K2, it could theoretically throttle the autoimmune damage while still letting the drug kill the cancer.
That is the zenith of precision medicine. But to bring this all full circle, we have to talk about how the drug alters the gut itself. This feedback loop is just mind-bending.
It really is. Because the gut has its own distinct immune cells, like innate lymphoid cells, specifically ILC3s.
And those gut-resident ILC3s express the exact same PD-1 checkpoint protein on their surface that the drug is targeting.
Right, so you give the patient an IV drug for a lung tumor, but it inadvertently hits these gut cells. It removes their brakes, causing low-grade inflammation in the intestinal wall.
Which brings us back to translocation, that localized inflammation causes the exact gut leakiness we talked about earlier.
Exactly. The drug causes the leakiness lets the Akkermansia translocate. The translocation primes the lymph nodes to send T cells to the tumor. It's a continuous closed biological loop.
The microbiome regulates the drug, and the drug structurally alters the microbiome.
It completely collapses the boundaries between medical disciplines. Gastroenterology, immunology, oncology, they're all one continuous operating system.
So final recap for you. Taking the brakes off causes autoimmune side effects, but different microbial signatures predict those side effects versus tumor shrinkage. And the ICB drug alters the immune cells living inside the gut, creating a massive feedback loop.
That's the whole picture. And the true paradigm shift here is that traditional oncology is fiercely focused on just treating the tumor, cutting it out, or radiating it.
Right.
But based on this cascade of evidence, shouldn't the very first step of cancer treatment be completely re-engineering the patient's internal microbial ecosystem?
It makes perfect sense. Why give someone $1 million immunotherapy if their gut is fundamentally programmed to resist it?
Exactly.
Which leaves you with an incredibly personal question to mull over. What is your daily diet currently programming your immune system to do?
Yeah.
If you could take a genomic snapshot of your gut microbiome today, what story would it tell about your body's defenses? Are you cultivating the diverse ecosystem required to fuel your immune assassins, or are you inadvertently clear-cutting the forest? Keep questioning the complex world inside you, and we'll catch you on the next deep dive.